At the end of the membrane story, we reached a problem. The plasma membrane can bend, pinch off, fuse, and rebuild itself, but it cannot organize those movements on its own. A phagocyte has to push its membrane around a microbe. A vesicle needs a route through the crowded cytoplasm. And a dividing cell must move two complete chromosome sets to opposite sides. The machinery that makes all of this possible is the cytoskeleton. The name can be misleading. A skeleton sounds fixed, solid, permanent, and largely unchanged. The cytoskeleton is almost the opposite. Much of it is continuously assembled, dismantled, moved, and rebuilt. It supports the cell, but it also allows the cell to change. That is the key idea to keep in mind. This structure stays useful because it never completely stands still.
Once you see the cytoskeleton as a working system, its major jobs become easier to organize. It positions organelles, establishes polarity so different regions can behave differently, generates movement, and provides routes for cargo. The cell does not use one universal fiber for all of this. It uses 3 filament systems. By diameter, actin microfilaments are the thinnest, roughly 5 to 9 nanometers. Intermediate filaments are about 10 nanometers. Microtubules are the thickest, at about 25 nanometers. Those measurements help you recognize them, but size is not the real reason 3 systems exist. The cell faces 3 different mechanical problems. It needs a network that can be rebuilt rapidly to change shape and generate local force. It needs tough cables that tolerate pulling and stretching. And it needs long polar tracks that organize the cytoplasm, move cargo, and rearrange during mitosis. Different problems demand different materials. Actin remodels, intermediate filaments resist tension, and microtubules provide long-range organization.
Let us begin with actin. Actin starts as a small globular protein called G-actin. These monomers join non-covalently to form filamentous actin, or F-actin. And 2 F-actin strands wind around each other to create a thin double helix. But the 2 ends are not equivalent. The filament is polar, with a plus end and a minus end. Under the right conditions, subunits are added preferentially at the plus end, while other subunits leave from the minus end. This is treadmilling. The important point is that the filament is not simply sliding forward as one solid cable. Individual actin molecules enter the structure at one end and leave at the other. The pattern can advance while the material itself is continuously renewed. That dynamic behavior is exactly why actin is so useful near the cell surface. The cell can build actin in one region, remove it somewhere else, and reshape its cortex rapidly. Now, place that dynamic actin directly beneath the plasma membrane. Here, it forms a dense network called the cell cortex. The cortex supports the membrane and helps determine the shape of the cell surface. When one region needs to move, the cell does not push its entire body forward at once. It assembles and branches actin locally. Growing filament ends press against the membrane and push it outward, creating a leading edge. The same protein can be organized differently elsewhere. Parallel actin bundles support a microvillus, while an equatorial actin ring appears during cell division. The principle is simple. build actin where the membrane must move, and local assembly becomes a local change in shape.
Polymerization can push a membrane, but pulling requires a motor. That motor is myosin. In a contractile network, actin filaments are arranged with opposite polarities, and a bipolar myosin II assembly bridges them. Using energy from ATP, each myosin head moves toward an actin plus end. Because the motors are linked, the opposing filaments slide relative to one another, and the network shortens. The actin molecule itself does not become shorter. The arrangement contracts because the filaments change position. Non-muscle cells use this logic for cortical contraction, migration, vesicle movement, and barrier regulation. During cytokinesis, actin and myosin form a contractile ring beneath the membrane. As that ring tightens, it deepens the cleavage furrow and separates one cell into two. Add a motor to a polar filament, and a structural network becomes a force-generating machine. Intermediate filaments solve a different problem. They are rope-like, relatively stable, and built to tolerate repeated pulling. Imagine one epithelial cell in a sheet receiving a local mechanical force. If that force remained concentrated at one membrane, the cell could tear away from its neighbors. Instead, intermediate filament cables attach to desmosomal plaques inside the cell. The matching desmosome in the neighboring cell links the two load-bearing systems across the junction. Force entering one cell can therefore spread through several cells and across the sheet. This network is not primarily a movement machine. It is a system for tensile strength and mechanical continuity. Its success is almost invisible. The tissue stretches, the load is redistributed, and the sheet stays intact. Stability gives intermediate filaments another useful property. Their protein pattern can preserve evidence of cellular lineage. Epithelial cells characteristically express cytokeratins. Mesenchymal cells commonly express vimentin. Muscle cells express desmin. Glial cells can express GFAP, and neurons contain neurofilaments. Lamins form a separate intermediate filament network along the inner nuclear envelope. Now, consider a poorly differentiated tumor. Its architecture may be so abnormal that morphology alone no longer reveals where it began. If it is found at a metastatic site, its location tells us where it traveled, not necessarily its origin. Immunohistochemistry can ask which lineage-associated proteins the tumor cells still express. A positive cytokeratin pattern supports epithelial differentiation and therefore supports carcinoma. Vimentin may support mesenchymal differentiation. Desmin supports muscle. GFAP supports glial origin. And neurofilaments support neuronal differentiation. But the correct verb is supports, not proves. Tumors may lose expected markers, co-express several markers, or show a marker outside its simplest textbook association. Vimentin alone does not prove sarcoma. The pattern becomes meaningful only when it is interpreted with morphology, a broader marker panel, and the clinical context. The third system is the microtubule. At about 25 nanometers in diameter, it is the thickest of the three major cytoskeletal filaments. Its basic unit is an alpha-beta tubulin heterodimer. These dimers repeat head to tail, forming protofilaments.
and 13 protofilaments close around a hollow lumen to produce one tube. That head-to-tail assembly makes the microtubule polar. Its two ends are not equivalent. In many interphase cells, minus ends are anchored near the nucleus at a microtubular organizing center called the centrosome. Plus ends extend outward toward the cell periphery. The result is more than a long rod. It is a track with a defined direction. A radial network of these tracks helps position organelles and organize the cytoplasm. Warmly, structure creates polarity, and polarity will soon become transport.
A microtubule is directional, but it is not permanent. At a growing plus end, GTP-bound tubulin is added and forms a stabilizing cap. The end may continue growing or pause for a time. If that stabilizing cap is lost, the protofilament tips curve and peel outward. The microtubule switches into rapid shortening. That switch is called catastrophe. If growth begins again before the track disappears completely, the recovery is called rescue. This behavior is dynamic instability, and it is not a defect. With minus ends anchored near the centrosome, populations of plus ends can repeatedly grow into the cytoplasm, withdraw, and try again. The cell does not need one rigid track to swivel like a compass needle. It continuously tests different routes by building and removing dynamic polymers. That lets the network explore space, reorganize quickly, and later search for chromosomes during mitosis.
The same tubulin material can be arranged into structures with very different jobs. During interphase, a radial microtubule network positions the Golgi apparatus, mitochondria, vesicles, and other organelles within the cytoplasm. At the cell's surface, microtubules can be organized into a cilium. A typical primary cilium grows from a basal body and usually contains 9 outer microtubule doublets without a central pair, the classic 9+0 pattern. It is generally a sensory projection rather than a beating one. Motile cilia use the classic 9+2 axoneme, 9 outer doublets surrounding 2 central singlets. Coordinated bending can move material across an epithelial surface, as in the respiratory tract. The sperm flagellum uses a related 9+2 architecture to move the cell itself. These are the standard teaching patterns, even though specialized exceptions exist. The main idea remains: change the architecture, and the same tubulin system can organize, sense, or move. Microtubular polarity gives the cell an internal coordinate system. In the common interphase arrangement, minus ends lie near the centrosome, while plus ends extend toward the periphery. Motor proteins can read that direction. Both kinesins and cytoplasmic dynein bind cargo through adapter proteins and use ATP to step along the microtubular surface. Most kinesins move toward the plus end, which usually means outward. Cytoplasmic dynein moves toward the minus end, which usually means inward toward the centrosomal region. That is a useful default map, not an absolute rule for every kinesin in every cell. The organizing principle is what matters. A polar track plus a selected motor produces a defined route. The cargo does not know its destination by itself. Adapters and regulatory proteins connect it to the appropriate motor. The same track can therefore support opposite journeys.
Direction matters because the cytoplasm is crowded. Small molecules can diffuse over short distances, but a large vesicle cannot leave the Golgi apparatus and simply hope to collide with the correct region of plasma membrane. A mitochondrion cannot rely on random drift to reach a region with high energy demand. Directed transport begins when an adapter loads selected cargo onto a motor. The motor engages a microtubule, walks toward the appropriate end, and releases the cargo at a defined destination. In the example here, a Golgi-derived vesicle uses a plus-end-directed kinesin to reach the cell periphery and fuse at the correct membrane domain. Mitochondria can also be repositioned along microtubules, often moving in either direction as local needs change. Transport may pause, reverse, or switch motors, but it remains regulated rather than accidental. A track provides the route, a motor provides movement, and an adapter gives the journey an address. Random drift is motion. This is delivery.
A neuron makes the transport problem impossible to ignore. Proteins and membranes are produced in the soma, on ribosomes and the rough endoplasmic reticulum, but the axon terminal may lie an enormous distance away. Diffusion cannot maintain that connection. In a typical axon, microtubule plus ends point toward the terminal. Kinesin, therefore, supports anterograde transport, carrying vesicles, proteins, and mitochondria outward from the soma. The terminal receives the material needed for membrane turnover, neurotransmission, and local energy production. The return journey is equally important. Cytoplasmic dynein carries used components, damaged material, and signaling endosomes retrogradely toward the soma. A signaling endosome formed near the terminal can deliver information back to the nucleus and influence gene expression. Without anterograde transport, the distant terminal is not supplied. Without retrograde transport, the soma loses both material and information from the periphery. Bidirectional transport lets one exceptionally long cell remain connected from end to end. When the cell enters mitosis, it does not keep the interphase transport network unchanged. The microtubule array is dismantled and rebuilt for a new job. Centrosomes have duplicated before this stage, and they now separate toward opposite sides of the cell. At the same time, chromosomes condense, and the nuclear envelope becomes discontinuous. New dynamic microtubules grow from the two spindle poles, some attached to kinetochores on chromosomes. These are kinetochore microtubules. Others overlap with microtubules from the opposite pole and form the interpolar region. Astromicrotubules extend toward the cortex and help position the spindle. By metaphase, these three populations form one bipolar mitotic spindle, with duplicated chromosomes aligned at the metaphase plate. The tubulin protein has not changed, but its architecture and purpose have. A radial organization system has become a chromosome segregation machine because dynamic tracks could be dismantled, redirected, and rebuilt around two poles.
Building a spindle is not enough. Every chromosome must be attached correctly before sister chromatids can separate. Dynamic microtubule plus ends grow through the spindle and search for kinetochores. When a plus end captures one, the first attachment may still be wrong or incomplete. Correct biorientation means that the two sister kinetochores connect to
opposite spindle poles. Pulling from opposite directions creates tension and helps stabilize that arrangement. Incorrect attachments are destabilized and tried again. The spindle assembly checkpoint is especially important here. An unattached kinetochore produces a wait signal that keeps the checkpoint active. Tension contributes to error correction and attachment stability, but the checkpoint is not simply a tension meter. Only when every kinetochore is attached and the chromosomes are correctly bioriented is the checkpoint satisfied. Then, and only then, is anaphase permitted. One unattached kinetochore can hold the whole cell at the line.
The spindle depends on controlled microtubule dynamics, and that dependence creates a therapeutic target. Taxanes bind polymerized microtubules and stabilize them. The tracks remain, but their normal remodeling is suppressed. Vinca alkaloids disturb the system from the other direction. They bind tubulin and interfere with microtubule assembly, reducing the functional spindle and promoting microtubule loss. These actions sound opposite. One makes microtubules too stable, while the other prevents normal assembly. Yet both remove the balanced turnover required for spindle formation, kinetochore attachment, and chromosome movement. The spindle checkpoint remains active, and the cell can arrest in mitosis. The target is not simply the presence of tubulin. It is the controlled dynamic behavior of the system. Push that system too far toward stability or too far toward disassembly, and division fails.
Now, look at the same disruption from the neuron's point of view. The problem is not simply one microtubule snapping in half. When microtubule dynamics and organization are disturbed, axonal tracks become less reliable and transport slows. Along the intact axon, kinesin carries vesicles and mitochondria toward the distant terminal, while dynein returns signaling endosomes and other cargo toward the soma. Both directions have to remain open. In the disrupted axon, cargo becomes stranded, fewer mitochondria and vesicles reach the terminal, and retrograde signals fail to return efficiently. The terminal is now undersupplied, even though the cell body may still look viable. Distance is the vulnerability. A peripheral axon may extend an extraordinary length, so a small transport defect is amplified across a very long route. That helps explain why taxanes and vinca alkaloids can produce a length-dependent peripheral neuropathy. The longer the route, the greater the dependence, and the earlier transport failure becomes visible. We now return to actin, but this time the whole cell is moving. Migration is not one forward shove. It is a coordinated cycle with a front, a rear, and 4 linked mechanical events. First, the cell polarizes. At the leading edge, actin polymerization pushes the membrane outward and creates a protrusion. But a protrusion alone can simply slide backwards. So the second step is attachment. New integrin-based adhesions grip the extracellular matrix beneath the front, and traction transfers force into that matrix. Third, actomyosin contracts through the cell body. Because the front is anchored, that contraction pulls the body forward instead of merely shortening the cell in place. Finally, adhesions at the trailing edge release, and the rear retracts. Then the same sequence begins again. Protrude, grip, contract, release. Notice that every step depends on front to rear polarity.
If attachment occurs in the wrong place, or the rear never lets go, local force does not become useful movement. Cell migration is therefore a cycle of coordinated asymmetry. Actin creates force. Adhesions give that force traction. Actomyosin moves the body, and rear release converts the entire sequence into net forward motion.
Cancer does not need to invent a new motor for invasion. On the left, a normal epithelial cell uses an actin-rich leading edge, integrin attachment, traction, and contraction to move into a wound. The movement is spatially controlled and remains part of a tissue response. On the right, a carcinoma cell reuses much of the same machinery, but normal control has been lost. Cohesion with neighboring cells is reduced, actin-rich protrusions extend into the surrounding matrix, and traction helps the cell cross the basement membrane. The machinery is familiar. The context is pathological. What changes is where movement occurs, when it stops, and whether tissue boundaries are respected. Invasion is deregulated normal movement, not a completely separate cytoskeletal program. A phagocyte can redeploy the same leading-edge logic around a particle. First, surface receptors recognize and bind the microbe. That local signal recruits actin assembly beneath the plasma membrane. As actin polymerizes, the membrane is pushed forward on both sides of the target. These advancing pseudopods form the phagocytic cup. The cup does not simply open and swallow. Its edges continue around the microbe until the tips meet. Membrane fusion then closes the opening and seals the microbe inside a new intracellular compartment, the phagosome. The phagosome membrane is derived from the plasma membrane, while the dense actin used to build the cup is largely dismantled after closure. Recognize, polymerize, extend, meet, and seal. It is the logic of an actin-driven leading edge, redirected around a particle.
The cytoskeleton can also leave a diagnostic record. Look at the tumor in the center. The cells are poorly differentiated, so their shape alone does not clearly reveal where they came from. Intermediate filament proteins can provide additional lineage evidence. Cytokeratins support epithelial differentiation. Vimentin is commonly associated with mesenchymal differentiation, although it is broadly expressed and not specific. Desmin supports muscle differentiation. GFAP can support glial differentiation, and neurofilaments can support neuronal differentiation. The important word is support. These stains are not perfect identity cards. Expression can overlap, tumors may lose expected markers, and some tumors express proteins that seem unusual for their apparent lineage. But a filament profile can still narrow an otherwise wide differential. That is why the central morphology remains ambiguous, while the surrounding stain patterns add direction. The pathologist is not asking which color names the tumor. The question is which combination of morphology and filament expression best fits a plausible lineage. Cell shape may become chaotic, but parts of the cell's molecular history can persist. Morphology may blur, filament identity can leave a useful fingerprint.
So, what does one positive stain actually tell us? Here, Cytokeratin positivity supports epithelial differentiation. It does not, by itself, prove the tumor type, and it certainly does not identify the primary site. Markers overlap, aberrant expression occurs, and technical or interpretive false positives are possible. A defensible interpretation comes from convergence. First, morphology tells us about architecture, cell type, mitoses, and necrosis. Second, an immunohistochemical panel asks whether several positive and negative stains form a coherent pattern and whether competing lineages are excluded. Third, clinical context adds the patient's age, tumor site, imaging, history, and other studies. These three streams enter the same reasoning funnel. The result is a supported interpretation, the most likely lineage and a narrower differential. It's stronger than one stain, but it is still evidence rather than mathematical certainty. That closes our first major cytoskeletal arc. The same system has moved cargo, built the spindle, driven migration, created treatment vulnerabilities, and preserved a diagnostic record. Keep the rules simple. A marker supports, a panel narrows, and neither should be allowed to prove a diagnosis alone. Now move from one cell to an epithelial sheet. The cells touch along the same lateral border, but they do not use one generic glue. 4 different junctions solve 4 different problems. At the most apical edge, the tight junction seals the paracellular route and helps keep the apical membrane distinct from the basolateral membrane. Immediately below it, the Adherens junction connects the actin belts of neighboring cells, allowing shape and force to be coordinated across the sheet. Lower on the lateral surface, desmosomes act as discrete spot welds. They connect to keratin intermediate filaments and distribute mechanical stress. Gap junctions do something completely different. They form small aqueous channels through which ions and selected small molecules can pass directly from one cytoplasm to the next. Notice the logic of the map. Tight junctions seal. Adherens junctions coordinate actin. Desmosomes resist stretch. Gap junctions communicate. The structures share one cell border, but each contributes a distinct tissue-level function. Let us magnify the first junction. Paracellular means between cells, not through them. Water and dissolved solutes approaching from the lumen enter the narrow extracellular cleft, where the tight junction controls whether they can continue. Claudin proteins form strands that bring neighboring membranes into close apposition. Their composition differs between tissues. Some claudins create selective pores that permit particular ions or water, while larger solutes and non-permitted ions are blocked. So, a tight junction is not always an absolutely closed wall. A very tight epithelium and a relatively leaky epithelium can use different claudin combinations to produce different permeability. What is prevented is uncontrolled leakage. Keep the route clear in your mind. The particles that pass remain outside both cells and move down the space between them. The tight junction turns that between-cell route from an open crack into a regulated tissue-specific pathway. The tight junction has a second job. One plasma membrane surrounds the entire cell, yet its apical and basolateral regions contain different proteins. The junction helps stop those membrane components from simply diffusing into one another. In this intestinal example, SGLT1 and peptide transporter 1 belong at the apical surface, where they face the lumen.
Glucose then leaves through GLUT2 at the basolateral membrane. The sodium-potassium ATPase is also basolateral. It pumps 3 sodium ions out and 2 potassium ions in, maintaining the sodium gradient that supports apical glucose uptake. The membrane fence restricts lateral diffusion at the apical lateral boundary. Polarized sorting and vesicle trafficking also help place each transporter correctly, so the junction does not establish polarity by itself. But once the domains are organized, the fence helps keep them distinct. That separation is what makes vectorial transport possible. Material enters from the lumen-facing side and exits toward tissue and blood.
Just below the tight junction, the Adherens junction couples the actin cortices of neighboring cells. The extracellular part is built around E-cadherin. E-cadherin on one cell binds E-cadherin on the next, and calcium stabilizes those extracellular domains. Inside the cell, the connection is not direct. The E-cadherin tail binds beta-catenin. Alpha-catenin and associated proteins then connect that complex to the actin belt. The same molecular bridge is mirrored in the neighboring cell. Follow the mechanical path across the board. Actin, catenins, E-cadherin, the extracellular cadherin bond, then E-cadherin, catenins, and actin again. A contractile force generated in one cell can therefore be transmitted across the junction and into the next cell. At tissue scale, these contacts form a continuous zonula adherens just below the apical seal. Because the actin belt is dynamic, the sheet can change shape while its cells remain coordinated. Warmly, cadherin provides the handhold outside. Catenins build the bridge inside, and actin supplies the force that is shared across the epithelial sheet. Desmosomes solve a different mechanical problem. They are discrete spot welds rather than a continuous actin belt, and they connect to keratin intermediate filaments instead of actin. Across the intercellular space, the desmosomal cadherins, desmogliin and desmocolin, bind the two cells together. Inside each cell, their tails enter a dense protein plaque. Desmoplakin in that plaque anchors the keratin filaments. Now, follow a pulling force. It reaches one keratin cable, enters the desmosomal plaque, crosses through the cadherin adhesion, and leaves through the keratin network of the neighboring cell. Because each cell contains several spot welds, the force spreads through many cables and across a broad region of the sheet. That distribution matters in tissues exposed to repeated stretch, such as epidermis and heart. A local pull is less likely to tear one membrane contact because the load is shared. Think of the desmosome as a membrane rivet tied to tough intracellular cables. One small weld, but a wide mechanical network. A gap junction does not mainly hold cells together. It connects their cytoplasms through a controlled aqueous pore. 6 connexin subunits assemble into one hemichannel, called a connexon. A matching connexon in the neighboring membrane docks with it to complete one gap junction channel. The two plasma membranes remain separate, but the aligned pore creates a direct path from one cytoplasm to the other. Ions, cyclic AMP, IP3, and selected small metabolites can diffuse through according to their electrochemical gradients. Large proteins and nucleic acids are excluded by size. The channel is also gated. During injury, a high cytosolic calcium concentration or low pH can close connexons and limit the spread of damaging signals into adjacent cells.
So the arithmetic matters. 6 connexins make 1 connexon. 2 connexons make 1 intercellular channel, and that channel permits selective communication without fusing the cells.
In cardiac muscle, that direct pour becomes electrical coupling. Depolarizing current in one myocyte passes through gap junctions at the intercalated disc and enters the next cell. If the neighboring membrane reaches threshold, it depolarizes as well, and the wave continues down the chain. Depolarization then opens voltage-gated calcium channels and triggers intracellular calcium release. Calcium binds troponin, cross-bridge cycling begins, and sarcomeres shorten. The gap junction did not pull the contractile proteins. It delivered the electrical event that activated them. Each cardiac myocyte still has its own membrane. Yet rapid ion flow lets separate cells activate in sequence and contract as a coordinated functional unit. Electrical coupling first, calcium activation second, coordinated contraction third. Step back now and look at the whole sheet. The cells remain separate, but 4 junction systems let them behave as a tissue. At the apical edge, tight junctions control leakage and help preserve polarity. Just below, Adherens junctions couple actin belts so cell shape and contractile force remain coordinated. Desmosomes connect keratin networks, spreading stretch through many cells rather than concentrating it at one border. Gap junctions pass ions and small signals, allowing rapid communication. Now, no single junction can replace the other 3. A perfect seal without mechanical coupling would still be fragile. Strong adhesion without communication would still be poorly coordinated. Communication without a barrier would not create a controlled epithelium. Together, the 4 systems produce the tissue-level outcomes shown here: a controlled barrier, stable shape, mechanical resilience, and a coordinated response. 4 structures, 4 jobs, 1 functioning tissue. Now, remove one part of that junctional system. E-cadherin normally gives neighboring epithelial cells a shared handhold. Its extracellular domains bind E-cadherin on the next cell, while intracellular adapter proteins connect the junction to the actin adhesion belt. That helps preserve a cohesive, polarized epithelial sheet. When E-cadherin is lost, that handhold weakens. Cell borders separate, the sheet becomes discohesive, and individual cells gain more freedom to move. On the board, that is the transition from an intact adhesion belt to loose strands and basement membrane crossing. But keep the conclusion precise. Loss of one adhesion molecule does not, by itself, complete invasion or metastasis. A cell still needs coordinated cytoskeletal remodeling, altered matrix attachment, basement membrane degradation, directional migration, and survival after detachment. So E-cadherin loss removes one important restraint on invasion. It can be part of an epithelial to mesenchymal program, but it is not the entire program. Once a cell leaves its neighbors, it does not enter empty space. It meets the extracellular matrix, and that matrix is not one uniform mesh. Directly beneath an epithelium is the basement membrane, a specialized sheet built mainly from a network of type 4 collagen, laminin adhesive glycoproteins, and proteoglycans. It supports and positions the epithelial cells. Deeper in the tissue,
The interstitial matrix contains thick rope-like type 1 collagen fibers, elastic fibers, fibronectin, proteoglycans, and stored signaling molecules. Both compartments provide mechanical information, including composition, stiffness, and tension. They also provide biochemical information through adhesive ligands and growth factors that can be stored, released, or presented locally. Surface receptors read those features and change intracellular signaling. That is why the matrix can influence survival, shape, migration, and differentiation. It is a scaffold, but it is never merely passive support. Its architecture and molecular composition form an information-bearing environment.
The receptor that lets a cell read much of that matrix is integrin. Each integrin is an alpha-beta heterodimer. The two subunits associate to form one receptor that spans the plasma membrane. Outside the cell, the head of the receptor binds a matrix ligand. Fibronectin is the main example here, and several fibronectin binding integrins recognize an RGD sequence within it. But integrins are a family. Different alpha-beta combinations recognize different ligands, including laminin and collagen. Inside the cell, the short cytoplasmic tails do not simply continue as a filament. They recruit adapter proteins. In a focal adhesion, talin and vinculin help connect the integrin complex to actin. The same platform recruits signaling proteins that influence survival, proliferation, migration, and gene expression. In another context, such as a hemidesmosome, integrins connect indirectly to keratin through proteins including plectin. So integrin is not just glue. It is a selective molecular bridge, matrix ligand outside, adapters and cytoskeleton inside, with signaling coupled to the attachment. Now, let that bridge do mechanical work. At the front of a moving cell, actin polymerization pushes the membrane forward. New integrins bind fibronectin and begin to cluster, forming a nascent focal adhesion. As talin, vinculin, paxillin, alpha-actinin, and other proteins assemble, the adhesion links those integrins to the actin system. Myosin then contracts the actin network. Through the focal adhesion, the cell pulls the matrix leftward or backward, while the matrix exerts an equal and opposite reaction on the cell. That reaction helps move the cell body rightward or forward. The front grip alone is not enough. Older adhesions at the rear must disassemble. If the rear remained permanently locked, contraction would stretch the cell between two fixed points instead of allowing productive advance. Cell migration is therefore a repeating mechanical cycle. Polymerize actin and extend the front, build a new grip, transmit actomyosin force, move the cell forward, then release the rear. Focal adhesions turn internal pulling into traction against the outside world. The same focal adhesion machinery also lets a cell test a physical property of its environment. Compare the same initial actomyosin pull on two matrices. A soft matrix yields. It deforms beneath the adhesions, so little sustained resistance builds across the integrin adapter-actin linkage. Focal adhesions tend to remain smaller and more transient, and the mechanical signal is shorter-lived. A stiff matrix resists the same inward pull.
Its outward reaction maintains tension, promotes the growth of mature focal adhesions and stress fibers, and can sustain intracellular signaling. Converting mechanical resistance into biochemical signaling is called mechanotransduction. Through this process, matrix stiffness can influence cell spreading, migration, survival, and differentiation. The exact biological response still depends on cell type, matrix composition, and signaling context. Stiffness is information, not a universal command. The logic is simple. The cell pulls, the matrix pushes back. Tension changes the adhesion complex, and that physical resistance becomes information the cell can use.
Desmosomes and hemidesmosomes sound like related versions of one junction, and both connect to keratin, but they attach a cell to completely different external partners. A desmosome is a cell-to-cell junction. Desmoglein and desmocolin are cadherins that bind across the space between neighboring cells. On each cytoplasmic side, an intracellular plaque containing proteins such as desmoplakin connects that junction to keratin intermediate filaments. Force can therefore pass from one cell through the desmosome into the keratin network of the next cell. If the junction fails, neighboring cells separate from one another. A hemidesmosome is cell to matrix. Alpha-6 beta-4 integrin binds laminin 332 in the basement membrane. Inside the cell, plectin and BP230 link the receptor to keratin. If this junction fails, the epithelial sheet can lift away from the basement membrane. So the shared theme is mechanical reinforcement through keratin. The decisive difference is the receptor and its external partner. Cadherin to another cell or integrin to matrix. That difference determines the plane of tissue separation. Integrins do more than hold the matrix. They also report what the cell is attached to. This direction, from an extracellular cue to an intracellular response, is outside-in signaling. First, a matrix ligand binds the extracellular head of an integrin. Ligand-bound receptors cluster in the membrane, bringing their short cytoplasmic tails together. On the inside, proteins such as talin and paxillin organize the adhesion complex, while FAK and SARC initiate kinase signaling. The signal then branches. One branch remodels actin and changes force transmission. Other branches, including PI3K-AKT and RAS-MAPK, influence the nucleus and gene expression. The possible outcomes include survival, migration, proliferation, differentiation, and a change in cell shape. The actual response depends on the cell and the surrounding context. The important logic is the direction. Ligand outside, integrin clustering at the membrane, adapters and kinases inside, then a cellular decision. What the cell is attached to becomes information the cell can act upon. Inside-out signaling runs in the opposite direction. The integrin is already in the membrane, but an intracellular signal changes how strongly it can bind outside. A bent, closed integrin has low affinity. Extension creates an intermediate state, and opening the headpiece produces a high-affinity receptor. Talin and kindlin both engage the beta cytoplasmic tail and stabilize this transition. Remember the distinction shown here. A conformational change raises the affinity of one receptor, while clustering many receptors raises overall avidity. Leukocytes use this switch during inflammation. A chemokine binds a G protein-coupled receptor on the leukocyte and generates an intracellular signal. LFA1 then enters a high-affinity state
and binds ICAM1 on activated endothelium, converting rolling into firm arrest. Platelets use the same principle. Platelet activation switches alpha-2b beta-3 integrin into a ligand binding state. Fibrinogen can then bridge activated integrins on two platelets and drive aggregation. No new integrin had to be synthesized. A signal inside changed the receptor outside, allowing a circulating cell to choose the exact moment to grip.
For many epithelial cells, correct attachment is not optional. Integrins bound to the basement membrane help confirm that the cell is in the right place. This is anchorage dependence. In the attached state, integrin signaling through FAC, SARC, and PI3K-AKT cooperates with growth factor signals to support survival. The matrix is therefore providing context as well as mechanical support. When the cell detaches, the alpha-beta integrin does not have to disappear or split apart. It can remain intact in the plasma membrane, but become unligated because it no longer contacts the correct matrix. FAC and PI3K-AKT survival signaling then falls. If the cell cannot replace that signal, pro-apoptotic pathways activate caspases. The cell shrinks, chromatin condenses and fragments, the membrane forms blebs, and membrane-bound apoptotic bodies are produced. This is anoikis, apoptosis caused by loss of appropriate cell matrix anchorage. It is orderly death, not explosive necrosis. The plasma membrane remains controlled rather than bursting. Anoikis removes a misplaced anchorage-dependent cell and helps prevent survival in the wrong tissue context. Anoikis resistance can survive detachment, but that only allows the metastatic journey to continue. It does not complete the journey. First, the cell must lose enough local cohesion, cross the basement membrane, and invade the interstitial matrix. It must then intravasate through endothelium into a blood or lymphatic vessel. In circulation, it faces shear stress, immune attack, and the absence of its original matrix. Anoikis resistance helps with only part of that challenge. Next, the cell must arrest on distant endothelium, extravasate into a new tissue, and survive a foreign biochemical and mechanical environment. Some cells remain dormant. A rare cell adapts, forms new integrin-mediated matrix contacts, receives suitable local survival signals, and begins clonal outgrowth. Compare the side branch. A normal detached epithelial cell undergoes anoikis and stops. The resistant tumor cell passes that checkpoint. But invasion, vascular entry, transit, exit, distant adaptation, reattachment, and proliferation must still succeed. Anoikis resistance is therefore not metastasis. It removes one barrier from a long, inefficient, multi-step cascade.
Now, step back and place everything into one functioning tissue. The membrane defines inside and outside and creates apical, lateral, and basal regions with different jobs. Actin supports the cortex and microvilli and generates force. Microtubules position cargo. Keratin intermediate filaments spread mechanical stress. Different networks, one cooperating internal system. At the lateral border, tight junctions control the barrier. Adherens junctions couple actin and coordinate shape. Desmosomes connect keratin and distribute stress. Gap junctions transmit ions and small signals. At the basal surface,
Integrins link matrix ligands to actin in focal adhesions or to keratin in hemodesmosome-like attachments. These contacts transmit force and biochemical information. The result is one connected physical system. Membranes define compartments. The cytoskeleton organizes space and produces force. Cell-cell junctions build a tissue. Cell matrix adhesions anchor it and tell each cell where it is. Every attachment carries position, force, and information. That gives us the next question. Where do these receptors, adapters, motors, and junctional proteins come from? Next, we follow protein production and routing through the rough endoplasmic reticulum and the Golgi apparatus.